we discovered a new type of cancer-causing gene in melanoma. A lot of oncologists are going to want this information because those results, they can mean one more season going to college football games together, or one more Christmas together, or an opportunity to see a child's wedding that otherwise might not have happened.
Hello, everyone. Welcome back to OncoDaily. And today, OncoInfluencers is honored to host a prominent figure in the oncology field, Dr. Levi Garraway. I don't think he needs much introduction. I'll leave the details to our discussion. Thank you very much, Levi, for having a time for us. When I was interviewing Philipp Kantoff and I asked who I should interview next, without thinking even a moment, he mentioned your name. Why?
Well, first of all, thank you, Gevorg, for the opportunity to chat with you. Phil has been a mentor and a friend for many years. He actually made some of the first phone calls to connect me to oncology faculty back when I was still a medical student. So I guess I'd say we all need mentors and sponsors like Phil. So he helped me a lot. And so I suspect that's maybe why I thought of me.
You have mentioned in the past that your family played a big role in your success. Can you tell us a bit more about it and how this influenced your career choices?
First of all, both of my parents had PhDs, which maybe in and of itself was unusual, certainly in the 1970s when I was growing up. But what that meant was their commitment to education was crucial to myself and to my two sisters, really for as long as we all can remember. But my parents, they set high standards, not just for academic performance, but also in how we live and how we treat each other. My father was a plant biologist, so he had a passion for science. That passion convinced me to pursue a scientific direction. I also, by the way, had two uncles who were surgeons, and one, his name was Levi Watkins Jr. He was a highly successful cardiothoracic surgeon at Johns Hopkins, and his career accolades in particular inspired me to become initially an academic physician scientist, but certainly the physician scientist element came from him.
You achieved so much in your career, but there was also a personal experience from which it all started. Please, could you tell us about the oncology choices as well?
Yeah, sure. Well, when I was in graduate school, my father was diagnosed with an aggressive form of prostate cancer. And really, up until then, I hadn't thought a lot about cancer. But from that point on, as my dad dealt with treatment and then relapse, and then there was just inevitable progression, and his ordeal really consumed our entire family. And at the time, I was doing a PhD in a very different area of biomedical research, but I soon found myself going to the library in the evenings and poring over the cancer literature, looking for some insight that might help my dad. But I have to say, those efforts, actually, not only were those efforts fruitless, they were quite frustrating because it was clear that there had been so many discoveries happening in cancer research. This is back in the mid-1990s. People were discovering oncogenes and tumor suppressor genes and major scientific advances. But that science at the time seemed to be having really a negligible impact on clinical outcomes. And that just felt unacceptable. So I'd say by the time it was clear that my dad was unfortunately going to lose his cancer battle, I had my own moment of clarity that I would want to dedicate my career to bridging the gap between science and clinical care in oncology, with the hope of bringing new treatment options, certainly in cancer and perhaps even in other diseases as well.
You have been a key figure in precision cancer medicine. Can you share your career story, how it started, how it went, and what was the moment in your career when you felt you were truly on the brink of something groundbreaking?
Well, I had the good fortune of obtaining my MD and PhD degrees from Harvard. I did my residency training in internal medicine at the Massachusetts General Hospital. And then I did a fellowship in medical oncology at the Dana-Farber Cancer Institute. By the way, I was also a chief resident in medicine at the Mass General, which turned out to be very important later in my career. But after that training, I did a postdoctoral fellowship with a guy named Bill Sellers at the Dana-Farber. And there we were applying new technology to ask questions of cancer genomes. And in doing so, we discovered a new type of cancer-causing gene in melanoma. That really paved the way for me to ultimately join the tenure track faculty at the Dana-Farber, at Harvard Medical School, and the Broad Institute. But the first moment where I really felt I was on the cusp of something groundbreaking occurred when I was just starting my faculty position. And at the time, it could cost a lot of money to generate a DNA sequence of even a single gene across a large number of cancer samples. And that was an important thing to do because we wanted to understand mutations of genes and their role in cancer. But at the time, I didn't yet have a lot of grant funding. So we were trying to figure out how to leverage technology to get the salient information from, cancer genetics in a much more cost-effective manner. And so we developed a method to profile the relevant areas of a large number of cancer genes, and to do that rapidly and cheaply. Actually, the term later would be called the actionable genome, but for us, it was just the actionable portions of a set of cancer genes. And we started finding potentially actionable mutations in many unexpected places. And at that point, I realized, you know what? A lot of oncologists are going to want this information to help them make treatment decisions as new targeted agents start coming along. And that discovery, that observation, became the genesis of a company called Foundation Medicine that we can talk more about later. But I would say it also inspired many similar kind of clinical genomic profiling initiatives around the world. And that was a key gateway to what we now call cancer precision medicine. But it all started back when we were trying to figure out how to do some relatively basic procedures in the lab in a more efficient and cost-effective manner.
What was the most difficult moment in your career, and how did you overcome it?
I can recall one time we had a very exciting. We were being funded to support a very exciting research project where we were running a clinical study looking at application of, at that time, now it was farther along, larger scale genomic characterization, and how can we use that to guide cancer precision medicine. And so this is about generating the data, validating the actionable alterations in a CLIA lab, using a tumor board to advise clinical decisions. But it turned out there were a lot of steps in that process. And once we started the study, we quickly became overwhelmed as the data was being generated. And really, the progress was being ground to a halt because we just didn't have the resources to just carry out this complex set of tasks. And so I remember calling a meeting with the entire team, and we had to confront the ugly facts that the project was failing. It was really collapsing under its own weight. And we actually had to make the difficult decision to suspend the study for several months so that we could get our act together and figure out how to rescue it. And I would say what came from that was, we had to own the challenges and the failures, but then at the same time, we had to get creative and think out of the box. How can we address the critical bottlenecks? What are the changes that are needed? And that creative thinking, together with some tough but necessary calls, allowed us to make changes that were needed and then to move forward and ultimately to successfully bring the project to completion. So it was a challenging moment, some difficult times, but sometimes those moments of adversity, they can force us to really consider what matters the most and to really invest our time and effort in the most salient needs.
What is the greatest source of inspiration for you today, and what keeps you moving forward?
Yeah, well, I would say my family, particularly my own kids, now helping them, not just my kids, but nieces and nephews, find their own path to success, hopefully learning from some of the same underlying principles that have guided my life, but also the next generation of physician scientists. I mean, they exude optimism, they exude resilience despite adversity that they're seeing. So it's always meaningful to carve out time for me, to advise and mentor, hopefully rising stars from the next generation. Those are a few things. For me, it starts with number one, finding work that you love and doing that work really well, focusing on being successful in that work, and not getting out too far ahead of yourself about what's going to be the next opportunity. Really focus on being successful in that work. Sacrifices and you have to work hard. There's no way around it. And being willing to find the time to put in those long hours, but then also to make sure to strike the balance and rejuvenate yourself. But then I would say it's also in doing that work and working really hard. It's also making sure that you treat people well along the way. Your colleagues, your friends, that you treat them the way you want to be treated, and then hopefully align yourself with mentors who embody the success that you're trying to achieve and who can help you. We all need help from other people along the way. So those are a few principles.
What do you hope your legacy will be in the world of oncology and cancer research? If we were to write a short sentence for your bio, how would you describe yourself in one sentence?
I would hope people would remember that my work enabled new cancer treatment paradigms and also multiple new cancer medicines that together brought a transformative impact to cancer care around the world.
Thank you. And going back to Foundation Medicine, can you tell the story, how it started, how it went? The story, which certainly will be very interesting for many people.
Yes, absolutely. So, as I mentioned earlier, there was the initial observation or set of exercises was rather mundane. We were trying in the lab to figure out how to get relevant genetic information from a subset of cancer genes much more rapidly and cheaply. And we assembled a technology platform that could do that. And it became immediately apparent that this could be useful broadly in the oncology community. And that really led to this overarching idea to create a single test that could read out genetic alterations in many types of cancer genes all at once. And then together, that information could be used ultimately to guide treatment decisions for new medicines that were emerging. So that was the idea. But of course, getting that idea to a product that could be made widely available was an entirely different issue. And I knew that I wasn't equipped to do that myself. So I reached out to some colleagues, including people at Dana-Farber and the Broad Institute, Matthew Meyerson, Todd Golub, and then eventually Eric Lander, who was running the Broad Institute at the time. And I remember sending Eric a business plan and meeting him in his office. And he basically said, great idea. There are a lot of obstacles to making it real. And somebody who was saying, really, you don't know how to do it, but I want to help you. And I know people who can help. And a couple of weeks later, Eric and I had dinner with some partners at a venture capital firm, Mark Levin, Bob Tepper, Neil Exter. And then together, the whole group that I mentioned, we proceeded to spend the next year and a half meeting every week, every Tuesday evening over dinner. In fact, it was my job to order the dinner, so I was uneventful. I was ordering from the same restaurant over time. They were very patient with me. But finally, after a year and a half, we felt like we had a plan to address all of the challenges that we could imagine about how you might take a genomic technology that was still emerging and turn it into something that could be used anywhere in the world by an oncologist. But by then, we had also become even more convinced about the opportunity. So the rest is history. We ultimately founded Foundation Medicine. It's now actually part of Roche, where I, of course, work. And well over 1 million cancer patients have now had a Foundation Medicine test performed as part of their care. And of course, there are many related cancer gene profiling tests available worldwide. So it really feels like this was enabling for cancer precision medicine in many ways.
Thank you very much. Going back to Roche, As the Chief Medical Officer at Roche, what do you see as the most significant challenges facing the global oncology landscape today?
Well, on the one hand, over the past couple of decades, we've made major advances as a field in targeted therapy, immunotherapy, and now with newer modalities coming along. But yet, cancer remains not only a global health burden, it's really still one of the leading causes of morbidity and mortality, and certainly in any local hospital or healthcare center, it feels like the patient examination rooms and waiting areas are still filled with patients who need treatment. So why is that? One challenge is that we have better treatments, but we're still not curing most people who have advanced cancer. And so what that means is resistance. Either the cancers don't respond at all to our treatments, and that's still true, unfortunately, for many cancers, or they respond for a while, but then they stop responding and then the cancer starts growing again. So that's acquired resistance. And these are major challenges still in many cancer types. So that's one major challenge is the issue of resistance to therapy. And related to that is there are still significant gaps in our understanding of tumor biology and in our ability to develop effective therapies for the biology that we do understand. So resistance and lack of the right biological understanding, these are still major challenges. Another challenge is a little different, but we could do better at diagnosing earlier and intervening earlier in disease where cancer can still be cured. So that's an area where hopefully new advances can help. And then lastly, kind of a bigger picture issue is these advances, they're exciting, but of course, they are incredibly costly to deliver. And so access and affordability to these advances, they remain formidable global challenges. So those are a few that are top of mind.
Can you elaborate on how Roche's approach to oncology differs from the other pharma companies in terms of innovation and patient care?
Well, at Roche and Genentech, as a company, we have invested heavily in oncology for a long time, for over 6 decades. And so as a result, there's a long legacy of therapeutic advances, new oncology medicines that have come from our company. And still today, we have quite a broad oncology pipeline. We have more than 30 right now new molecular entities really across an array of not only cancer types, but also modalities, small molecules, large molecules, gene therapies, new types of cancer immunotherapies, et cetera. So we also have not just this large pharmaceutical R&D effort, but we also have a large diagnostics division, all as part of the same company. And that's unique in the industry. And that enables us to advance cancer care really across the entire patient journey, from screening and early detection and more accurate diagnosis and molecular subtyping, and then all the way into treatment and monitoring. So I think that's a unique element. And then finally, I would say our areas of oncology R&D focus, we have a balance to address both opportunities for near-term advances. For example, we have a number of ongoing trials in breast cancer and lung cancer and hematologic malignancies. So there's a balance of those near-term opportunities to those opportunities that may take a little bit longer to play out, such as modalities like allogeneic cell therapy or new therapeutic types like cyclopeptides, which we're doing in collaboration with Chugai, which is a Japanese company that's part of the Roche group. So when you put that all together, we have a long history, a broad pipeline, pharmacologic and diagnostic effort together, and then a balance between nearer term and longer term focus. And I think together that makes us uniquely positioned to continue to make advances for a while.
And I'd like to ask also a question about your mentors and mentees. Well, I know one already. I'm fortunate to know him, but I'd like to explore more about your mentors and mentees.
Absolutely. Well, the first thing that I'll say is that there's a long list of amazing men and women. And in fact, if I give a talk and try to mention career mentors, actually it requires 2 slides. So I won't be able to mention everyone, but it starts with, we all over the course of our career, there are people at various phases. So when I go back to even when I was a graduate student and postdoc learning how to do science, my PhD advisor, Steve Beverly, and my postdoctoral mentor, Bill Sellers, they were such rigorous, critical thinkers. And how do you design the right experiment? How do you think about the right question? So I have to start with them. And then there are other broader career mentors that I had early in my training. People like John Potts, who is at Mass General, amazing leader. Phil Kantoff, who introduced me to you, and Ravi Thadani, who was a clinical investigator and teacher for residents. They profoundly shaped my early career directions and thinking. And then once I landed my faculty career, people that I've mentioned already, like Eric Lander, Todd Golub, Matthew Meyerson, such great mentors and colleagues during the academic research years. And then as I was making a transition to leadership or to industry, people like Sue Mahoney, who was at Eli Lilly at the time, or Lori Glimsher, who became president of Dana-Farber. I learned so much about leadership from those individuals. And then I would say today, there are a series of current and former C-suite leaders from the companies that I've been involved with and institutions that I remain close to. So the bottom line is there are so many mentors, and the single most important thing that ties them all together is that there's nobody telling them they don't have to mentor me. They choose. They chose to dedicate their time to help me to be better or help me to find my way. And so that's it's to the extent that we can all take that to someone, whether it's a trainee or a team member or really anyone who asks. That's part of how we pay it forward and make the world better in the space of biomedical science.
Thank you very much. What would be your advice for the younger generation, for those who are just starting their career and who want to be successful?
So the first thing I would say is hang in there, keep the faith. This is a special field where special advances that change the world are possible and sometimes even right around the corner. That's one. The second is to identify and focus on the most important work. And what I mean by that is younger generation, or when you're just starting out in the field, there are always lots of opportunities that come around. And the ability to be ruthless to say no to work that's nice to have, but not must have. Because when you say no to work that's nice to have, but not must have, you have found time that you can now dedicate not only to the most important work for making that discovery or that treatment advance or that biological advance, but also to other things, like things in life that matter even more, such as your family, your relationships, even hobbies that give you joy. So really being mercenary about focusing on the most important work. And then finally, it comes back to this fundamental principle of you find work that you love and then do it well. Focus on excellence in the work that you are doing. And when that happens, you don't have to go searching for opportunity. Opportunity will find you.
Continuing the same line, what would be the books you would recommend that everyone reads, both in the field and outside?
When I think about books that I've read in recent years that have been highly influential to me, they're not books about cancer necessarily. And so one book is Good to Great by Jim Collins. And from that, I learned practices and behaviors of leaders that were able to bring their organizations to an unprecedented achievement. So that's certainly a book worth reading. Another book that I read more recently is called The Power Law. It's by Sebastian Mallaby. That book deeply influenced my thinking on what transformative impact looks like in general, and then what does that look like in medicine or medicines that we're trying to develop. And then a third, actually, there's a four-volume tome by Robert Caro called The Years of Lyndon Johnson. And I would say especially the third volume, Master of the Senate. That book taught me how to get things done in highly complex organizations. And also it's just a sobering reminder about how the difference between one path or another path in history profoundly affected by, sometimes unlikely individuals or events. So those are 3 books that I would focus on.
Last 2 questions. If not medicine and science, what would be your choice?
Probably teaching. Teaching, hopefully in a scholarly institution, to shape the ability of the next generation to think originally, but also critically.
And the last question, which I usually ask everyone, who I should interview next?
I would recommend Charles Sawyer's or maybe Arul Chanayan. And I would say, well, one thing that has kept me going over a period of years, and especially in my current job, is that there's just nothing quite like seeing the Kaplan-Meier curves, the survival curves that show separation of cancer patients who are now living longer on a new treatment. And knowing that I had a significant role in moving a treatment like that forward, I mean, that's a major motivation. Or seeing a waterfall plot that's showing tumor shrinkage in patients who might otherwise not have had a chance to get the treatment had we not made the call to do the clinical trial. Those types of results, they never get old. And I just, every time I see a result like that, I always harken back to my own experience when my dad was going through his own cancer struggle, because those results, they can mean one more season going to college football games together, or one more Christmas together, or an opportunity to see a child's wedding that otherwise might not have happened. And so those are motivating. It's also true, drug development is not for the faint of heart. Disappointments when it turns out that you were maybe not quite right about the therapeutic hypothesis, those can be heartbreaking for the patients and for the families and for everyone involved. But the successes, those positive studies where you see the curve separating and people living longer, those, they can change medicine. And when you can do that, that keeps you going through all kinds of adversity.
Thank you very much, everyone, for being with us. This was Levi Garraway.
Thank you for the opportunity.
Don't forget to like, comment, share, and subscribe to OncoDaily on YouTube. Hit the bell icon to stay updated.